on review of our meiosis and mitosis. We're going to be completing a lab today. Our goals are going to be to make sure that we understand our mitosis and what is going on with the DNA as it moves through each of the phases. So, we're going to begin with a warm up. Our warm up is going to be for you guys to list the stages of mitosis. So, I want them in correct order. Okay? If you guys need any help, make sure you guys refer back to your ISNs, your interactive student notebooks. So, I want on the top of the paper, I want you guys to put stages of mitosis and then go ahead and number them. You should have six total, okay? I do want you guys to include interphase and cytokinesis. Alright, and then when you guys finish, put your pencils down so I know that you guys have finished. Alright, excellent. Eyes on me. Alright, so, Jason, what's the first stage of mitosis?
Interface.
Very good. Nick?
Prophase.
Very good. Chelsea?
Metaphase.
Very good. Sherry?
Anaphase.
Very good. And what's the fifth phase? Everybody?
Telophase.
Good. And our last one is
Cytokinesis.
good. And cytokinesis is right, kind of considered a part of telophase. Also, it's kind of on its own thing. So, very good. So, if you're going to break it down into six, those would be the six, right, for the whole cell cycle, including mitosis. Okay. So, as we go through, we're going to watch a small video, just kind of quickly reviewing our stages of mitosis. So, can everybody see? All right.
During the mitotic phase, a cell will undergo mitosis to form two new nuclei, and then divide to form two new individual cells during cytokinesis. Mitosis is the process of dividing the duplicated DNA of a cell into two new nuclei. Mitosis is split into distinct stages. The first stage is prophase. The DNA condenses, organizes, and the classic chromosome structure appears.
All right, that's our X shape. That's our
Comes
chromosome.
prometaphase, where microtubules
All right.
attach
We don't have to worry about this yet. That's a little bit
This
beyond it.
step is followed by metaphase, where the chromosomes align.
Okay, remember they're lined up in the
Metaphase
middle.
is followed
Middle's
by
metaphase.
where the chromosomes separate.
Anaphase pulling apart.
Finally,
Anaphase.
during telophase, nuclear membranes reappear around the two sets of chromosomes. Mitosis is now complete. After mitosis, two new cells are formed by a process called cytokinesis.
Cytokinesis, where they're pinching
Mitosis
off.
is only one part of what is called the cell cycle. For many eukaryotic cells, a cell is duplicated every 24 hours. Most of the life of a cell is spent in interphase. Interphase consists of three stages called...
All right. I'm not going over interface today. All right. So just a quick little review over our mitosis. So what we're going to do today is we're working on a lab, okay? So here's our materials. Okay, you guys are going to be partnered off, okay? So I'm going to have you two be a partner, and I'm going to have you two guys be a partner, okay? So this big guy right here, okay? This guy's going to represent our cellular membrane. Okay, see, it's clear so we can see in it, see what's going on. Okay, so this is going to be our cellular membrane. Okay, our rubber bands, these are going to represent our nuclear envelope, our nuclear membrane. Okay, our quarters. Okay, quarters are going to represent our centrioles. And then finally we have paper clips. Okay, these are going to represent our chromosomes, right? They can kind of make an X. They're linked together. Okay, so we have different colored chromosomes to help us kind of track where our DNA is going throughout our stages of mitosis. All right, and then finally we have our two daughter cells, right? As mitosis goes on, right, the goal is cell division, so it's going to produce two identical daughter cells with identical DNA. All right, so each group will get their own set. Okay, so there you go. You guys got yours. All right. So we're gonna start out in interphase. Okay. So I want you guys, as a team, to produce. Okay. I want you guys to produce interphase. All right. So go ahead. Okay. So you guys are gonna need your nuclear envelopes, your nuclear membranes. All right. So I want you to tell me, where is the DNA? Jason, where's the DNA?
Inside the nuclear envelopes.
Okay. And then our centrioles are just kind of floating around, correct?
Yes.
All right, very good. So, on your warm-up sheet of paper, I want you guys to label and then draw interphase. Okay, so on the bottom half, okay, draw a line. And you guys are going to draw a circle. Then I want you guys to draw label interphase and then draw a picture of what you see. Yeah, make sure you make it big enough so that way you guys can label it. Okay, you guys need to be able to make sure you know what goes where. All right, so Nick, make sure go back to your ISN. Okay. The chromosomes, they don't form until we get into prophase. Interphase, the DNA is just kind of floating around. The chromatin's just kind of floating around. Okay, so it's just kind of squiggles right now. Not X, it's just squiggles. There you go. Yep. Yep, perfect. All right, then when you're done with that, go ahead and put your pencils down so I know you're done. Very good. All right, so what is our next phase in mitosis? Our first phase of mitosis, what is it?
Prophase.
Prophase, very good. Okay, so with your cell, I want you to go ahead and move your cell into prophase. Okay? Right? It should look very similar. Now, what's going to happen is our nuclear envelope, as you can see, starts to disappear. So go ahead and get rid of our nuclear envelope. Okay. The chromosomes are going to stay in a tight bunch. They're kind of still going to be close to each other, but our nuclear envelope disappears. All right, very good. Now, again, go ahead and draw what you guys see. Very nice. So that's where it moves into the chromosomes. You get that normal X shape that we're used to looking at. All right, everyone's done? Awesome. All right, so our next phase is what?
Metaphase.
Metaphase, correct. So, what is the key identifying thing that we're looking for in metaphase? What happens?
Chromosomes line up.
Awesome. Yes, the chromosomes line up in the middle, right? Middle, metaphase, the M. Okay, so go ahead and move your cell into metaphase now. Okay, we're going to line up those chromosomes. Okay, you can have them in an X or just in a line, that's fine. Either one works for me. All right, line them up kind of towards the middle. And what happens to those centrioles? Where are they going?
The sides.
Right, they're going to start drifting towards the sides. Okay. Good, so the centrioles are going to start drifting off to the sides. Perfect. Very nice. Okay, so this is, we don't have spindle fibers in this particular demonstration, but this is where the spindle fibers would start to reach out. Okay, they're going to start reaching out towards the chromosomes. They're going to grab one chromatid, right? They're going to grab one chromatid from each side, because our next phase is going to reach out with those spindle fibers and grab them. All right, and what is our next phase?
Anaphase.
Anaphase. Now, what's the key to anaphase?
The
Right.
chromatids are splitting into singles.
They're pulling apart, right? So our chromosomes are going to be split apart, and they're going to become chromatids. Okay, those spindle fibers are going to rip them apart. So apart, anaphase, right? They go together. Anaphase, apart. Okay, so go ahead and move our cells now into anaphase. Okay, so you have two. Good. You have two linking paper clips. Okay, two linking paper clips. You're gonna unlink them. Very good. So now we have a chromatid of each represented, and they're going to be pulled towards our centrioles. Okay, so they should be. You should have one chromatid for each centriole. Very good. All right, so again, go ahead and draw what you see there. Okay, if you need help, refer back to your ISNs. Okay, refer back to your notes. All right. Very good. And so, our last phase is going to be what?
Telophase.
Telophase, right? Because telophase is kind of incorporates cytokinesis. So, let's demonstrate telophase right here. So, what is with telophase? What do we notice that's different? Okay, it's still kind of one. It's still one cell. There's still one nuclear membrane, but what do we see? That's what starts coming back.
They're starting to split.
envelope.
Oh, the
So-
envelope.
Our nuclear envelope. Very good. Okay, so now we need to incorporate our nuclear envelope, and what goes inside our nuclear envelopes?
The
The
chromatids.
chromatids.
Right, the chromatids. Okay, so inside you have two rubber bands. Okay, so you have two nuclear envelopes, and you're gonna have good. Right, because our just one. I just want one. No, that's cytokinesis. We're still on telophase. Okay. So, very good. right? Our centrioles, they don't go inside our nuclear envelopes. They kind of stay on the outside. Our DNA, that's what stays inside our nucleus. Very good. Okay, so draw what we see. Okay. Now, if we could make this perfect, right, these would be a little bit more drawn out, a little bit more elongated. Very nice. Very nice. All right, so into our last phase. Our last phase is going to be what?
Cytokinesis.
Cytokinesis. Okay, right? It's could be considered part of telophase. Sometimes it is. Sometimes it's considered its own phase. All right, so cytokinesis. All right, and what's our major defining moment of cytokinesis?
The daughter cells.
The pinch off, right? It pinches off into two daughter cells. So move our cell into cytokinesis for our last part of mitosis. That's it. Perfect. Great job. All right. Now I want to ask you guys, okay, as you guys are drawing, go ahead and draw. Okay. I want you guys to think about, so what would these new daughter cells, what would they move into now? Right? What's the next part of that cell cycle? What would they move into?
Interphase.
Interphase. Very good. Right? So they'd move into interphase. They would spend most of their cell life in interphase, and then they themselves would then go into mitosis themselves, splitting into, again, new daughter cells. Okay. Right? And so remember, with these pictures, our DNA is going to start becoming unwound, right? You're not going to see our chromatids, okay? You're not going to see our chromosome shapes. You're not going to see the chromatids. They're going to kind of just be flowing around. The DNA is going to be a nice long strands. All right, awesome. Do me a favor, go ahead and pick up all our equipment, put it back in the way that you found it. Okay. Okay, you don't have to connect the paper clips, just put them back in the bowls. All right, awesome, very good. All right. Now, for our wrap up, okay, we have a review sheet for mitosis. Okay, now what it has is it's got the picture, and I need you to name what the picture represents. Off to the side, it says what is happening. So give me the key thing. What is the main thing that separates this phase from the other phases? Right? What is that key phase? What's the key thing that's happening? Okay. Again, if you need help, refer back to your ISN. Okay. It has your notes inside. All right. If you need help, go back to your ISN. Okay. All right. You guys can talk to each other. Ask if you need help. Ask your partner. Okay. It's okay to collaborate. Talk. Okay. What do you guys think? What do you think is the main
Metaphase, anaphase, and then telophase. Yeah, it's not using the last two that we went over here. Yeah. Metaphase.
Good. But what envelope? It says the envelope is disappearing. Which envelope? Okay. so while you guys are wrapping up, go ahead and keep on working. All right, what do you think the first phase is? What's the first phase? This
Prophase.
Prophase.
Prophase, very good. And what's the key identifying mark?
The nuclear envelope
Is what?
is fading away.
Very good, right? Nuclear envelope's fading away, and you're going to start to see our DNA is going to start forming into our chromosomes. Okay, so what's the second phase that is listed here?
Metaphase.
Okay, and what's the key identifying mark?
The chromosomes start to line up in the middle.
Chromosomes line up in the middle, right? That's a real easy one to see. When we move on to our microscopes, okay, that's going to be the one that most of you guys are going to be able to see the easiest. Okay, you're going to see that nice clean line, okay, inside those cells. All right, and the next phase would be
Anaphase.
anaphase. And what's, again, the key identifying mark?
The chromatids split away from each other.
Good, the chromatids split away, all right? When we get to the microscopes, this one will be also a little bit easier to see. Okay, you're going to see kind of groups of the purple strands are going to be pulling away. Okay, there's going to be a small split in between them. And then our final phase is,
Telophase.
all right, very good. And our key identifying mark?
The
The nuclear
splitting.
envelopes reappear.
All right, good. Our nuclear envelopes reappear. Okay, you're going to start to see a pinch in that middle. Okay, that's also one. And then not listed is cytokinesis. All right, very good. All right, well, we ran out of time today for our meiosis portion. All right, but let's go ahead and let's pack up. Let's put away our materials. Very good. Make sure we put them in our ISNs so that way we don't lose them. Awesome, and you guys are dismissed.